It's with profound honor and great enthusiasm that I welcome you all today to what promises to be a truly inspiring and intellectually enriching occasion. The academic community of the Warsaw University of Life Sciences is privileged to host a scientist whose contributions have revolutionized molecular biology and genetics, a pioneer whose work has reshaped the way we understand gene expression, and those discoveries continue to open new frontiers in medicine and biotechnology. Please join me in warmly welcoming our distinguished guest, Professor Craig Mello, Nobel laureate in physiology and medicine. I would also like to warmly welcome Ms. Alina Pszczółkowska, AstraZeneca Country Vice President. It's also my pleasure to welcome Mr. Adam Smith from the Nobel Prize Outreach. Ladies and gentlemen, Professor Mello, together with Professor Andrew Fire, was awarded the Nobel Prize in 2006 for the discovery of RNA interference, a fundamental mechanism for gene silencing. This breakthrough not only deepened our understanding of gene regulation, but also ignited a new era in biomedical research, enabling the development of innovative therapies, from antiviral drugs to potential cancer treatments. For a university devoted to life sciences, your presence today, Professor Mello, is not only a celebration of scientific excellence, but also a powerful reminder of the curiosity, perseverance, and imagination that drive discovery. Your journey from the lab bench to the global scientific stage exemplifies what is meant to challenge established thinking and to ask bold, transformative questions. To our students and early-stage career researchers, today is a rare opportunity to learn directly from one of the greatest minds of modern science. I encourage you all to listen carefully and closely, to engage actively, and to let yourself be inspired, because the future of science is not just shaped in Nobel lectures, it is behind the conversation and questions that follow. Professor Mello, thank you for joining us today and for generously sharing your time and insight. We are deeply honored by your presence. I would like to welcome now Ms. Alina Pszczółkowska, the Vice President of AstraZeneca Country, to the stage to give a short speech and just a short organizational comment. I very, very deeply ask you not to use mobile phones during the lecture. Thank you very much.
Your Magnificence, Rector, esteemed Vice Rectors, Chancellors and Deans, dear scientists and dear students. It is with the great pleasure and honor for me to introduce and welcome today Professor Craig Mello, the laureate of Nobel Prize, and the great scientists. I don't think I can even imagine how the Nobel laureates feel, how proud they feel when they receive this recognition, not only because of the recognition itself, but primarily of the impact their work and discoveries has on the world. What Professor Mello did were thanks to the discovery of the RNA interference mechanism and the gene silencing was two things. One, it is about the pushing boundaries of science in the genetic fields, but the other is to open the door for the treatment of many diseases and impact the humans' lives. I think this is as far as you can get in the science, not only change the scientific paradigm, but also impact the people's life. We are here today to learn more about the Professor's Mello discovery and work, but we are also here to get inspired. And I'm especially touched by the number of students being here with us today. You are the future of the Polish science. And it can be that we have the future Nobel Prize laureates in this room today. So I hope that today's lecture will inspire you to think bold and bring your vision and your exploration to life. There is no coincidence that we are here in one of the best Polish universities because the life sciences like biology, biotechnology, they lay the foundation for the future discovery in the medicines. It is the academia when the ideas are born. But there is another step which I personally feel inspired and engaged in. And this is the collaboration between the private sector and the biopharmaceutical companies and the academia, because this is the steps when the scientific idea can be turned into the real medications which can get to the patients. So let's get inspired today. Let's get inspired for the future discoveries, the breakthrough discoveries in the science, but let's also get inspired for the collaboration so we can change the world together. Let me welcome on the stage now Professor Craig Mello. We are looking forward to the lecture today.
Thank you all. It's such a pleasure to be here and to get to know the Polish people a little bit, to see this beautiful country for the first time. I have to say I'm very, very impressed and happy to know that you're here. It's just wonderful to see the enthusiasm for science that your society clearly has, and the beautiful institutions you have for education. It's also wonderful that to be here as a guest of AstraZeneca. This program that they sponsor has had a tremendous impact all over the world, sort of giving an opportunity for people like myself to meet you and to share how exciting this, whether it's biology or chemistry or physics, just how beautiful and exciting the world of research is right now. The talk I'm going to give today is about information. And I gave it this kind of creative, crazy title, RNA interference, a molecular spark in an information inferno, because living things are really all about information. Information makes life. That's what the living state is driven by, this struggle for survival among informational molecules. And so that's kind of the theme for the talk, this struggle for existence among self-replicating informational molecules, this fire, if you will, of information that began on the Earth billions of years ago is still happening today and driving living the complexity of life. This movie is taken by a colleague of mine, Jim Priest, showing the beautiful movement and dynamics of egg production in this tiny little animal, C. elegans. C. elegans is a worm. I'll introduce you to it a little bit more in a moment. But it is also a very close living relative of humans. Bob Horvitz calls the worm a tiny little human. And they really are. We're very closely related to them because we shared 3 billion years of common ancestry with this animal. And about 1 billion years ago, maybe a little bit more, our ancestor was an egg being formed in an ovary very much like this. And right next to it was another egg that went on to make the nematodes. We had a common ancestor with this animal that made both lineages, the human, the vertebrate, and also all the invertebrates. These animals on the planet today all had a common ancestor, bilateral symmetrical animals like ourselves. They're very similar to us. They have a nervous system like us. They have a musculature like us. They have metabolism like us. The green stuff here that's flowing beautifully into these forming eggs, that's mitochondria. They used to be bacteria. Now they're part of us. We are a blended organism. The information from the bacterial life form that gave rise to mitochondria is part of our information. It's moved over into the nuclear genome that's inside the dark circles in this movie. That's the nucleus of the cell. That's where the DNA resides. The mitochondria have their own DNA, but it's only a fragment of what used to be inside the mitochondrial genome. Thousands of bacterial genes have been transferred precisely into the nuclear genome of us and of this common ancestor that I'm imagining here. That's a living worm right there, obviously. But our common ancestor engulfed, became symbiotic with, lived together with, assimilated an entire genome into our genome. It became a blended organism. This is the horizontal flow of information from one group that evolved independently into another group of living things. This happens constantly. It's the horizontal flow of information, not from parent to offspring, but across species, jumping from one organism to another. Genes move constantly. This is the theme of, and kind of the learning of years of working on information and how organisms handle it. I got interested in this question when I was a little kid, 17 years old, reading the newspaper, and I saw this article saying that a human gene had been put into bacteria, and that bacteria could read the human genetic code and make human insulin protein. And I read that article with growing fascination because I could not believe it. After billions of years of separate evolution, which I had learned about from my dad, who's a paleontologist, how could it be true that the language of the cell, of the human cell, was the same as the language of a bacterial cell? How could that be true? And it really captured my imagination, in part because the whole article that I read, and I read every word with fascination, it did not once mention that isn't it amazing that the genetic code is the same in bacteria and humans? Think about it. That is amazing. And why is it true? There was no explanation given. There was no scientist quoted saying, isn't it amazing that the genetic code is universal on this planet? And it practically is. There are only tiny little differences out there. Why is that true? Think about it. Oh, and people, I've realized that people really love the simple explanations, and maybe that's why, because it's so complicated, they didn't want to talk about it. This is just kind of a funny explanation of RNA interference, and it came on the nightly news in the United States where they really have to boil it down and make it simple. This is a picture of double-stranded RNA, and those are defective genes that are flying into it. It's the Pac-Man model for how RNAi works. The reason I'm showing this is because when we try to explain things, we really often oversimplify them. In some cases, we make up things that are just not true. RNA does not chew. It doesn't do that. But part of it is this human, this just sort of human reaction to oversimplify. Living things are incredibly complex, especially when it comes to how they handle information. And I'm going to tell you a little bit more about how RNA interference really works, but it's been quite an adventure, because when we first started working on this, we had no idea of what we were getting into in terms of how this really incredible system works. And I'll say more about this in a moment. But first I want to just show this simple introduction to RNA, and this is going to bring everybody up to speed on what RNA is if you don't already know.
Every creature, and you know this from high school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different for mice, and then for whales, and for flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big.
So I know you guys probably all know what RNA is, but if you don't, it's the cool stuff. RNA is the program that's running in your cells. DNA is like a hard drive that stores stuff. When you want to open an application and you want to make things happen, you have to express your RNA. And that is where really the fun starts. Now, RNA is also a really interesting molecule because it's somewhat reactive. As many of you know, it can actually catalyze. It can be an enzyme. And RNA, we believe, was the beginning of life on the planet. Way over on the left, you see this, what we call the RNA world, origin for molecules that could self-replicate, essentially beginning this struggle, this fire of information, this struggle to be present in the next generation. Over time, these molecules became more sophisticated. This is all, what we think. It's hard ever to really know for sure. Although I imagine someday I'll put some of these bullet points up in case I don't mention them. Eventually, maybe we can simulate this, or who knows, explore a new world that has life. And we'll learn more, certainly, from having another example of it. But the genetic code is universal on this planet. And the reason it's probably universal is because these organisms, this molecular life form that arose on the planet, never died, and it inhabits or moves horizontally, and essentially the information flow is driving the sort of the maintenance of this code. In other words, the code is the same because if you can't speak the language, you'll be at a huge disadvantage because there's, useful information arising in one lineage can be captured and used in another. And I mentioned this already with mitochondria. Chloroplasts are the same way. Organisms share information all the time. Over evolutionary time, information flows horizontally, which means that organisms have to have really sophisticated ways of handling information. Information, if you don't know it yet, it is dangerous. We are all learning that lesson because information is now at the fingertips of every human being. How do you know which information is safe, which information is true? Organisms have been dealing with this problem for billions of years. And our genomes, the human genome and the worm genome, look really weird because they're packed full of repetitive DNA, of things that look like the remnants of viruses. 30% of our DNA looks like retroviruses, like HIV. The genome is full of evidence of previous insertion events. And these elements are still moving around. This fire never went out. These informational molecules are vying for survival by interacting and propagating themselves inside of our DNA. This is truly fascinating, fun aspect of biology. It's a whole layer of natural selection, evolution that is unappreciated. Maybe a few of you in the room work on transposons, but they are super cool. Most of our DNA is transposon DNA, and they drive a lot of our biology. So really fascinating stuff going on here. RNA interference it's so ancient, the mechanism of RNA interference, that it was present in the common ancestor of bacteria, fungi, and plants and animals. And one of the things that even a lot of biologists don't appreciate is that the mechanisms that underlie living things are really so sophisticated. All organisms, people often say higher organism and lower organism. There is no such thing. Everything alive today is at the pinnacle of evolutionary complexity. Everything alive. There are no simple living organisms today. And a lot of what we think of as progressive evolution, the evolution of us, we think of as, oh, we're so much better, we're bigger, we're smarter, etc. Really, below this ice, the Earth was covered by ice all the way to the equator about 640 million years ago. There were multiple episodes of this in the history of the Earth. And most recently, around 640 million years ago, below the Cambrian, living things under that ice sheet already had RNA interference. They had a nervous system, they had bodies, they made eggs. There were we know this from genetics and genomics, really sophisticated organisms were alive on this planet, but nothing could live on land. There wasn't even any soil because the Earth was glaciated right down to the equator. So, it's really interesting to look at the history of life, and you see this amazing explosion of diversity after the Cambrian. This is really, really telling us something. It's telling us that organisms are incredibly capable of adapting to local environments, and that when the Earth warmed and oxygen levels increased, living things rapidly evolved to become big. But the tiny little things, like the worm that I work on, they are just as sophisticated as we are. And this movie actually shows this snake-like organism. That is a single-celled organism. It is called a ciliate. And they have argonauts too. I'll tell you more about argonauts in a moment. But look at these things. That's a single-celled animal. And the way that it gets big is by cutting up its genome into fragments and expanding every copy of every gene. And that little animal crawling in the middle, that's a metazoan. That's a multicellular animal. Really looks kind of clunky compared to the beautiful single-celled animals. Those animals coordinate the movement of those cilia so precisely that they can swim and hunt and eat. They have a mouth. I mean, think about it. These are tiny, tiny little animals, and they use argonauts to guide they use RNA to guide the entire remodeling of their entire genome every life cycle. They chop their whole DNA into tiny pieces. Each piece has one gene on it on average, and they amplify their genes 10,000-fold so that inside one cell they have 10,000 copies of every gene, or 30,000 copies even. And that's how they get big. Instead of cell division, they amplify their genome, but first they fragment it into pieces. This is incredible. You guys, really, it's incredible. Living things are super cool and really, really incredible. And here's the real Nobel Prize winner, this little worm called C. elegans. It is, well, now 8 gold medals and 4 prizes from the Nobel Committee. This is an amazing animal. I'm going to have to slow down. I like your audience. You guys are great because I see people smiling in the audience. I love it. I feel like I'm connecting with you, but I really want to get to your questions. So I apologize for being so enthusiastic that I will probably talk for hours about, my favorite little organism or the history of life, and I'll never get to actually what the heck is RNAi. It's really a cool story. Here you're seeing a worm getting injected with DNA. This is how Andy Fire and I got to know each other. We were trying to figure out how to put DNA back into the animal. This was before I got all excited about RNA. I didn't realize at the time that RNA was so much more important than DNA. I thought DNA was cool, so I was trying to put DNA back into this animal, and we had to figure out how to inject them. Here you see a needle injecting DNA solution directly into the ovary. And you can see that better in this picture, where you can see that the ovary is essentially a common cytoplasm with multiple nuclei that are surrounding it. And so you can put a needle into the worm, you can flow in DNA, you can flow in a CRISPR mix, you can genome edit this animal so easily. I can make any nucleotide substitution I want in this animal using this technique called CRISPR. You've probably heard of it. It works fabulously well in this animal. I can change any single nucleotide easily, and 3 days later I can have my edited animal. I can model a human disease very simply. I'm not going to bother explaining all this, but these are examples of GFPs that were made by CRISPR, can knock in any gene you want. It's a great system. So it's like a dream come true for me. CRISPR is amazing. Genome editing is such a powerful tool. It allows humans to make precise mutations. But what is RNAi? Now, RNAi was something discovered very accidentally. Nobody really predicted it would exist, but in the early 90s, people working with C. elegans, and my good colleague Ken Kempfuse in particular, described really strange silencing response to injected RNA. And they were injecting RNA the same way we did for DNA. In fact, I think I taught Ken how to inject the DNA after we finally figured out how to make it work. You have to inject the DNA into the germline, into the gonad. If you don't inject the DNA into the gonad, into the ovary, you never get a transform. You never get DNA in the progeny, right? So to get DNA to be inherited, it makes sense, you have to inject the DNA. Oh, so anyway, that's DNA. RNA, however, surprisingly, we discovered quite by accident that you can miss the gonad. You could just stick the needle anywhere in the body and inject double-stranded RNA into the animal, and it would silence genes, not only in the injected animal, but also in their progeny. We had this incredibly surprising phenomenon, purely phenomenological. Lisa Timmons and Andy Fryer's lab actually showed that you could modify the bacteria the worms eat, and these animals are so small that they eat bacterial cells like M&Ms, one at a time, and they actually crunch them in like a pharynx. I wonder if they taste good, I don't know. But basically, she found that and she didn't even tell Andy she was doing this, she put a gene into bacteria and transcribed it in both directions to make double-stranded RNA, targeting a worm gene, and she showed that just by feeding them that bacteria, the animal would mount a silencing response, and it would silence that gene. And so when in 1998 we published this observation, along with the fact that you could get inherited silencing for multiple generations, we published that in a Nature paper. We couldn't believe it got accepted. We submitted this paper that had absolutely no mechanism at all. It was all phenomenology, but it was so unbelievable. It was so incredible. I guess we got lucky. We had reviewers thought it was cool, but at the time, it was kind of hit or miss. One person you describe it to, they get all excited. Another person you describe it to, they look at you like you're full of, whatever, malarkey. But the bottom line is, we really didn't know what was going on, but we knew it was cool. And we had a great way to find out what was happening. We could ask the organism. We could use genetics. So the way you do that in C. elegans is you grow up a population, you treat it with a mutagen, and then you can do a genetic screen. In this case, it was really easy using Lisa's observation. Hiroaki shown here, engineered bacteria to express a gene required for embryogenesis. So if the worms feed on this double-stranded RNA, they inactivate an essential gene, and the embryos die. It's amazing, but it works. And so he could do this incredibly simple screen. He could feed them bacteria and then look for worms whose embryos don't die. Those worms would have a defect in the RNAi machinery. And he did the screen, and it worked so beautifully. The next year, 1999, we already had identified hundreds of mutants because it was a selection. It was really beautiful. And it was super, super cool. Really, really cool. And I got so excited about this, even before we cloned the gene, we submitted a paper because a lot of the mutants activated the transposons. The things I told you about, the junk DNA, the repetitive DNA, the little things that are not really junk. They're actually doing all of these informational things in our germline, in our genome, rewiring over evolutionary time, maybe, some people think. The transposons became active when you knocked out genes required for RNA interference. But that wasn't all. That was cool. That was super cool, and we were so excited to find that. But what was even more cool was that there were other mutants where RNAi did not work at all. It was completely wiped out, but the transposons were still silent. And that told us that double-stranded RNA triggers RNA interference and shares a mechanism, but probably a different trigger that maintains transposon silencing. Unlike the CBS model for RNAi, it was already getting complicated. It meant that it wasn't double-stranded RNA only that was initiating silencing, but that there was some other mechanism, some other cue, some other trigger that was maintaining transposon silencing. And I thought that was so cool. I put it in the title of the paper. And of course the paper got rejected because it was too complicated. They didn't want this complication of, oh, you know, some genes are required for transposon silencing and some are not. And then, anyway, long story short, it continued to get more and more interesting, more and more complicated. But fortunately for us, we cloned RDE1, one of these genes that is not required for transposon silencing. And what we found is that it has a whole family of genes it's related to, including human genes, genes that are in all of us. The worm had 26 homologs of RDE1. And RDE1 was required for the double-stranded RNA response, but not for the transposon silencing. And when we looked at the family tree, we saw that there were 4 human genes, the little HS means human, that were very similar to a couple of worm genes, ALG1 and ALG2. And then there was another set of human genes over there in green, 4 of them, that were very similar to a worm gene, PRG1. So the common ancestor of worms and humans already had 2 different groups of genes. This is the most simple explanation anyway for what you see here. It's a family tree, and it suggests that humans and worms had a common ancestor that already had several, probably, Argonaut proteins, which is what we now call them. And of course, RDE1 was required for RNAi, and we immediately began to ask which of these other genes are required for transposon silencing. So we began, with help from our collaborators, to systematically knock out every one of these genes and try to assign them functions. And this was where really the fun began, and it got more and more interesting, more and more complicated. We knocked out ALG1 and 2, and we needed to knock them out both together because they're redundant. You knock out one, they're fine. Knock out the other, they're fine. Knock them both out, the animals die. And interestingly, when they die, they look like a mutation that had been previously described in the microRNA gene LET7, which is a lethal gene encoding an RNA which Victor Ambros discovered the first of, called microRNAs, that form naturally into a double-stranded shape by forming what's called a hairpin. The RNA folds up and makes a double-stranded helix in the middle. We were not all that surprised at this point, but we kind of expected it, but we were thrilled that we had actually found a genetic connection between the RNA interference mechanism, the response to exogenously provided double-stranded RNA, and the microRNA pathway, which had been discovered in 1993, a few years before our work showing that we could trigger silencing by giving animals double-stranded RNA. Organisms have been using microRNAs since the common ancestor of worms and humans to regulate gene expression. In fact, the LET7 microRNA is identical in the worm and the human at every single nucleotide. And it regulates some of the same targets. Worms and humans are closely related organisms. This is why all of you should be working on C. elegans, including AstraZeneca. I'm working on them. I'm going to keep working on them. Anyway, worms have so much more to teach us. Worms actually have a big amplified group of argonauts called the Wago argonauts. And I'm not going to go into all the details, but it really is cool. It's all published, most of it, but it's so complicated, probably none of you will, even if you work hard at it, you probably won't appreciate it. I hate to say it, because when we write the papers, often we don't even understand it ourselves. But the Wago argonauts, this expanded group of argonauts, they help to propagate the silencing from one generation to the next. So what are argonauts? They're proteins. This is an example from a structure determined by Dinshaw Patel's lab. showing how beautiful they are. Proteins fold up and can do these amazing things. In this case, this protein can hold on to a guide RNA, which is shown here in red. It holds tightly at one end, and it shields most of the RNA. This is from really nice work from Ian McRae and other structural biologists, and it presents the guide RNA to targets. Oh, I haven't shown it yet. This is Google. It's a search engine. It's programmable search. Organisms they have been alive in the information age for billions of years. They handle information really, really cautiously. This enzyme can find matching information by presenting the guide RNA a few nucleotides at a time. If the first 3 nucleotides match, more nucleotides are exposed. Ultimately, if all of the nucleotides match, you get 2 turns of the helix inside of this protein, which can then position the target in blue for cleavage. And this thing operates at 100 million collisions per second, near the diffusion limit. It prevents the RNA from sticking to stuff unless the RNA matches. It's actually slowing down base pairing interactions, making sure that only the precise matches are found. This is how this guided search mechanism works. It's also very similar to how other guided search like CRISPR works. CRISPR is an RNA-guided DNA nuclease. Argonauts are an RNA-guided RNA nuclease, and they're fundamental to life. None of us would be here without this mechanism. It's also very fundamental for our germline. And we can make drugs using this now. I'm taking way too much time. It's fun though, isn't it? It's like, this stuff is so cool. You cannot make this up. It is so surprising and so amazing. A few programmed Argonauts in a cell can shut down every gene, every mRNA expressed in that cell easily. mRNAs are long, they're slow to make. Argonauts move fast, they target, they're catalytic, they can turn things over. And it just kept getting more and more complicated. And a lot of the stuff here on the right is unpublished still, but when you start working on silencing, you find out later that you're actually working on licensing, because for everything that's turned off, there has to be some way to keep things on, because, obviously some genes need to be expressed. You can't silence them all. There's this whole really other cool thing here going on. Of course, we were able to show, along with work from many other labs, that piRNAs regulate transposon silencing. So indeed, there's a separate Argonaut, humans have one too, that regulates transposons. And this is, of course, happening right now in all of, certainly every male germline. This is where the peewees are expressed primarily in humans, but they are regulating transposons in humans and in worms. And it's a very ancient gene family, really interesting. And in both the worm dsRNA response and in the transposon silencing pathway, worms engage in RDRP, which is an RNA-dependent RNA polymerase that humans do not have. But this is an ancient RDRP. Plants have it. Many different animals have it. Also, fungi have it. But humans have lost it. RDRP amplifies the silencing. This is why worms are so good at silencing, and it loads another Argonaut. So downstream of both RD1 and the piRNA pathway, there's another Argonaut engaged. That Argonaut includes an Argonaut that goes into the nucleus. That Argonaut can recruit chromatin silencing and establish heterochromatin on the genome. And in every eukaryotic, practically, not everyone, but most, including plants and animals, heterochromatin and small RNA pathways are tightly linked. And they together work over in the germline over multiple generations to select an off state and maintain it. Interestingly, there's, in the worm in particular, an argonaut that seems to play some role in anti-silencing. Really cool. I mean, you cannot make this stuff up. And I kid you not, I'm not dumbing this down at all. This is so complicated that even in my field, even the people who publish in this field do not believe what I'm telling you. They don't believe it, and I don't blame them. I didn't want to believe it. I mean, I was forced to believe it because, experiments kept working out the way they did. It's like, that's the fun part about science. Sometimes it drags you along a path, sort of kicking and screaming. Anyway, there's another silencing pathway shown here that humans also have, interestingly. It's called the HUSH pathway, the human silencing hub. And it detects defects in mRNA splicing, which is this really crazy thing that happens when many protein coding genes are expressed. They get spliced, pieces get chopped out, and ends get joined together very precisely. You end up with a message that's shorter than the genomic copy of the gene. That's splicing. If you mess splicing up, or if you're a retro element and you make a long RNA that's never spliced, there's a pathway that detects absence of splicing. It works in humans. We discovered it in worms recently. We didn't know it was related to this human pathway. People working in the human discovered this other pathway, the HUSH pathway, and we tested this gene out, TASER1. It's not an argonaut. It's not even related to an argonaut, but it seems to be playing a role in sensing defects in splicing. In the worm, it senses defects at the 5' splice site, and it triggers silencing, which leads to heterochromatin formation, which leads to the loading of argonauts, which leads to small RNA silencing. And so some of the mutants that we work on, also the ones downstream, the WAGOs, for example, end up important in this pathway as well. And there's another splicing sensing pathway that's TASER independent, where we haven't identified any genes yet. It detects defects in the 3' splice site. Again, there's probably layer after layer of this, and we're beginning only, I believe, to scratch the surface of the various mechanisms that our cells are using to sense gene expression and make sure that it's safe. I just wish humans would be more careful with information that we are ingesting, right? If we would just vet that information and be more careful, maybe a lot of things would work out better for our own society. Information is so key, so fundamental, and so important to all living things, and yet it is also incredibly dangerous. And so they have incredibly elaborate mechanisms for controlling it. The information is even passed in the sperm of C. elegans, and these little granules you see surrounding the sperm nuclei, these are filled with argonauts that are transmitting a silencing signal from the male to the progeny of that male. Worms have amoeboid rather than flagellated sperm, so you can see it crawling around. Anyway, Adam, how am I doing for time?
To be honest, bad.
We've got
another 5 minutes.
Okay, in the interest of time, and because I really love having a chat with you guys and with Adam, I think I've set the stage pretty well. There are medicines already based on this. We can talk about them. It works in humans. You can trigger this in humans. It was hard, but the drugs now are already working. They're getting better. They work great in the liver. My colleagues have broken open brain delivery. There's a company developing siRNAs for Alzheimer's and a variety of neurological disorders. There's even an siRNA in human trials for preeclampsia, a condition of pregnancy. siRNAs are incredibly safe. They're very durable. You give an siRNA to a human, you can knock down a gene for 6 months. The gene stays down for 6 months in the liver. The data looks really good in the brain. We think we can dose patients maybe once a year to treat Alzheimer's and many other disorders. We can use microRNA like siRNA technology to regulate genes that are master regulators of metabolism. We can make you fit, strong, healthy. We can help your bones be stronger. I bet we can even make you more attractive, more handsome, better looking, curlier hair. I don't know what you want. But yeah, I mean, gene expression, it's really beautiful that we can now make drugs based on genetic mechanisms that are natural, that are at work constantly, that are highly effective, that are very safe, and in this case, completely reversible. Once the drug is gone, the patient's gene expression would go back to normal. Admittedly, not every disease can be treated by knocking a gene out. That's what siRNAs do, but we can talk more about how you can use this as a therapy. So in the interest of time, and I didn't even get through any of like the stuff, you know, I kind of said some of the stuff, but I'd love to tell you more about some of the fun stuff going on in the lab. But in the interest of time and to give you guys a chance to ask your questions, I'm going to stop there and invite Adam to come up and let's do a little dialogue. Shall we? All right, thank you. Okay, where do you want me?
You want to go sit there?
Here? Okay, okay. Good. Well, he works me really hard. I have to tell you, I love working with Adam, but I don't think I've done so many interviews in 2 days in my whole life. Well, maybe since the Nobel.
I'm truly embarrassed to have been, yes, the cause of really Craig's non-stop routine since he arrived in Warsaw. I wanted to thank you so much for giving all this energy to the discussions today, the discussions yesterday. It's been an absolute joy to watch you and to talk with you. And I also very much wanted to thank SGGW for welcoming us back again. It's such a pleasure to be here. Thank you very much indeed. This program is very special to us, and it's really wonderful to see all your faces. Thank you for being here. Craig, your enthusiasm for all this is so palpable and infectious. It's just lovely to watch. And I'm sure that people will want to ask about that joy in the science. I wanted to pick up on one thing just to start before we open up for questioning. You said you didn't want to believe the results just now. And that's quite interesting because young scientists are taught, or young everybody is taught, that science proceeds by testing hypotheses. And you might think that you just have an idea of how things will be and you're trying to test that that's right, and you're basically trying to prove yourself right all the time in science. But you really emphasize that that is not how things proceed.
It's true. I mean, yeah, everybody likes the explanations to be simple, right? And there's this principle that I'm sure you're familiar with of Occam's razor. The simplest explanation is always the best. And so you naturally try to come up with your, simple model. I admit I never came up with the Pac-Man model. I just laughed so hard when I first saw that CBS News actually aired that model. That is just so funny. But yeah, I mean, it just kept getting more and more complicated. And in fact, it still is. There's just still so much more, and there's so many more layers, and I love that. I mean, I do love it, although it's humbling, because you do have to constantly admit you're wrong. That's the fun thing. Science is really interesting. I do believe it's a unifier. It brings us all together, because when you're doing science, it really is humbling. And and you gather information in an effort to test your models. And in fact, it never proves you're right. It can only, the data can only disprove your model, really, you know? And that's kind of the beauty of it. You use information, you gather it in order to try to disprove what you believe. And if you can disprove it, then you learn something. So it's a good thing, right? That's the only way humans ever learn anything, is by disproving what they thought they knew. And it totally flips around how you're supposed to use information. Instead, what most people do is they seek out information that confirms what they believe. But I think the scientific method really is the healthy way and the only way to safely handle information, which is in an effort to disprove your beliefs. And that needs to be more widely accepted by society. I think we need to teach people that lesson very, very much so now, especially considering that you can fake almost anything now. And so, yes, I think we have a lot to learn from nature, but also just from the scientific method. And it's not just scientists who do this. You know, I think we have a long ancestry as a curious creature, that our culture and our evolution, everything we do, all of the skills that we've developed culturally, agriculture, domestication of plants and animals, these were all done by the same method, the scientific method. It's the only way humans have ever learned anything. I love it. It's very simple.
It's very important. Thank you very much. I really would love to have questions from the audience. So we have 3 microphones in a row there. Please don't hesitate to stand up and come stand in front and ask a question. Don't be shy. This is your chance. Please. Yeah, please.
The microphones are there. Just jump up and walk to them. We've already got one, but please.
Please, at the back.
The more
yes.
the better. And you can ask anything you want.
What's your favorite color?
That's a hard one.
Yeah. Thank you very much for this excellent talk. You hinted a bit the formation of the P granules, and now we appreciate more that RNA contributes to various sorts of condensates. So my basic question is, what do you think is the actual unit of inheritance of small RNAs, and could some sort of higher order structures be actually transmitted between the cells and through the cell division?
Yeah, I mean, I didn't get to talk about the beautiful P granules. That was a few slides more. They were actually, I even forgot to mention them in the movie, didn't I? The movie showed them in that beautiful green movie where the mitochondria, I pointed that out. There were these beautiful little red balls, you probably noticed them, surrounding each nucleus. Those were the P granules that the question is about. The P granules are these beautiful liquid-like condensates that are so beautiful that when I showed them to my mom, who's an artist, she said, "Craig, what are those?" And I said, "Mom, nobody knows." And she's like, "You should work on that. That is going to be important." And the funny thing is that about 20 years later, we found that that's where the Argonauts are in the germline. A lot of them are in those beautiful domains called nuage. They're really fascinating, and we're working on them constantly because trying to figure out what they are. But it seems like the Argonauts are positioned there because the transcripts, the RNAs that are going to be expressed, are exiting the nuclei, and they have to navigate through the nuclear envelope, and then they immediately encounter the nuage where the Argonauts are sort of poised to screen them and detect, what might be dangerous information or to regulate them. So yeah, they're really, really fascinating. I have a bunch of stuff relating to them. It's one of the things that's really exciting about them is each individual domain of nuage, we believe, and we have a little bit of evidence supporting this, we think is different in that we think that they're servicing different transcripts because the heterochromatin is positioned around the nuclear periphery, This is getting into the weeds a little bit, I apologize. But it's really cool because when we look at them, we can see that different nuage domains have different transcripts in them. So it looks like they become differentiated. And in fact, we think that they're more like nucleated by the interactions between RNA binding factors and the target RNAs. And I didn't get a chance to show it, but we have evidence that the RNAs can even be aligned inside the nuage, 3' end at one end, 5' end at the other. And I have some images for that. We haven't published it yet, but we're working on a paper on that. It's really cool. Again, I'm sorry because this is part of the problem with it. It gets so complicated. Journals hate it. It's hard to publish this stuff, actually. People want something simple. And unfortunately, my experience is living things are complicated, you know. And you probably know this. I sense, you know, one of the folks here, you're probably one of them who works on these things. They're cool.
I know that it's impossible to encapsulate, but do you have a candidate for the unit of inheritance?
RNA is clearly guiding inheritance. I mean, I think it's absolutely clear in the ciliates, especially. The ciliates, I showed you one example of a single-celled animal in that movie. There are many, many different types of ciliate, but the things that they do with their argonauts in terms of remodeling the genome of these nuclei that are, oh gosh, I'm sorry for the jargon, but you guys are, some of you are mostly scientists maybe, but the jargon does get in the way. But in the ciliates, RNA guides every generation. The RNA guides something that is akin to DNA splicing. The DNA gets chopped up into bits and reorganized by an RNA-guided mechanism. So in the ciliates, it's pretty clear that RNA in some ciliates is guiding the assembly of genetic information every single generation. I don't think RNA ever gave up its role as the informational molecule of the cell. I think probably every generation, transcription of all the genes in the genome somehow plays a really important role in inheritance, and that's probably why it's so complicated. I mean, living things are really, really sophisticated, and they have complicated mechanisms. I'm not embarrassed to admit it. You know.
I think it's really fabulous to see how you dwell in the complication. I mean, you get so much done, so much comes out. And as well as all this, you produce companies that are, you know, heading towards therapeutics and all the rest of it. But how do you actually get anything done? Because you're constantly distracted by this immense detail.
It is hard, and it actually, you know, but it's fun, and I enjoy it. I mean, I feel bad sometimes for my students because, you know, it's easy to get a PhD if you solve a problem, you know, but if you just uncover more complexity, your PhD starts to be really, really hard because you can't wrap it up. You can't, you know, like I've had students spend 6 years in the lab and not get a single publication, and that's not right. But they still get a PhD and they deserve it, but it doesn't look good on their record, you know, so I feel bad about that, and it does happen. We don't reward people for tackling really hard problems. We reward people for solving easy problems, you know, and that's not good. I mean, because the hard problems, you know, they deserve to be solved, and trying to solve them, you might actually fail. I mean, and does that mean you're a failure? No, it just means it's a hard problem. And we don't reward people for that. I think there should be extra credit for people who take on something really complicated and don't solve it, but maybe show that it doesn't work one way. You know, it's hard to publish that stuff.
That's such an important point. And of course, if it does work, you get a Nobel Prize. So.
Yeah, exactly. The upside is pretty big, right?
Next question. Sorry, you're going. Yes, please. It's fine. Please come up.
I have a simple question. What is this anti-silencing? Could you tell us more about this? This is something really fascinating. Thank you.
Yeah, that's a really, there's no short answer to that. That Argonaut engages guide RNAs that are derived from the genes that are not silenced in the germline. So there's an RDRP that essentially templates, meaning uses as a transcribes RNA from RNA. It makes an RNA matching the genes that are expressed. Those RNA guides are loaded onto this Argonaut Caesar-1. This Argonaut is an essential Argonaut, so it's been hard to study. When you knock it out, the embryos arrest, and even the chromosomes fragment at the first division of the embryo. So really important Argonaut, essential, hard to study. But it's loaded, it's programmed with guides that target genes that are clearly not silenced because they encode proteins, including Caesar-1 itself, that are not silenced. And we use genetics to show phenomenologically in genetic crosses between strains that had a silent gene and and another strain that had an active copy of the same gene, that in some crosses, the active copy somehow interacts with the silent copy. These are two independent lines. Let's, for simplicity, just say it's a GFP tag. You cross them together, and there's a transitive effect in which the active gene transitively activates the silent gene. It's transactivation. We published this. This paper is seldom cited, and yet no one has refuted our data. I totally stand by the data. We have shown that in that instance, this genetic cross, that this Caesar-1 Argonaut is required for the transactivation of that silent gene. And we also know that in the strain where the gene is active, the GFP sequences, which are here, I didn't explain any of this, so I apologize to all of you who don't know what I'm talking about. GFP is green fluorescent protein. This is from jellyfish. It's a foreign gene. When you put the foreign genes into the animal, it somehow knows they're foreign and silences them. The pyrene pathway does this. It's super, super cool. Anyway, somehow the worm strain that has it on, the GFP on, has adopted that as a self gene because Caesar-1 is loaded with small RNAs targeting GFP. And when they cross them together, anyway, new data, totally new data. We think we're going to be able to finally get people to accept this result. We now have evidence that the organization of the mRNA inside the nuage is, oh God, I can't explain this. Trust me, it's cool. It's really cool. We have new evidence that during each life cycle, the gene that was undergoing silencing is retranscribed at a time when it's not normally going to make a protein. It's being transcribed in order to increase the silencing signal. So they transcribed at this early stage in the germline, in the young larva, the germline is transcribing or turning on the genes that were silenced in the last generation. And during that event, we can get it to happen synchronously because when the larvae hatch, they are in a state of suspended animation until you feed them. When you feed them, they synchronously begin transcribing all their genes, and they also begin transcribing, anti-intuitively, they begin transcribing the genes that were silenced in the previous generation. Those transcripts from the silenced genes accumulate inside the nuage, the P granules, where they form linear arrays with the 3' end at one end and the 5' end at the other. The silenced region, the region that's undergoing, that we triggered the silencing on, is in one domain of nuage where RDRPs reside and amplify the silencing signal. The other part is in another domain of the nuage where the RDRPs are not present. And they're bundled, they're multiple RNAs that are there probably for this amplification process. And when you knock out the Caesar pathway, the compartmentalization fails, all of them collapse together. So instead of being segmented this way, the different domains of nuage mix, and we think it's sort of spatially disturbing the amplification mechanism and preventing. I think it's super cool, but again, you can't make this stuff up. It is so weird, and I love it because of that, but it's fun.
And do you think it only happens in C. elegans or also in other organisms? Is it
Oh
like-
yeah, that's been our worry, keep it short, for the longest time. But no, I think it's probably universal,
Thank you.
although different in different species.
I think for those who are close to the subject, that was very enlightening. And for those who are a little bit more distant, the thing to see is that getting a Nobel Prize doesn't in any way make you less hungry to know how things work. I mean, in your case, it seems to have increased your hunger to understand. And that's important to see, that you're just desperate to know how. Because at the heart of being a great scientist and a Nobel laureate is that you really need to know the answer. It's not that you kind of, sure, it's all interesting. It's that you absolutely have to answer this question in front of you. That's, in a way, all of you, all Nobel laureates, that's what you have in common.
I think it's also a lot of luck. Do you have a question?
Hello, Professor. I just wanted to know the therapies with this that is being drugs that are being developed. They also trigger autoimmune responses. Yeah. So how is this being studied? And I also wanted to know, because if you want to study these systems in plants, we have to spray with such RNAs. Time to time, it's not very stable. And how are these drugs stable inside a human body?
Great question. And the answer to the first part about stimulating autoimmunity, one of the serious problems with most RNA drugs or even DNA-based drugs is when you give DNA or RNA to an organism, you can get an immune response that in the human can be very potent, and it causes flu-like symptoms, inflammation, even cell death. And so RNA and DNA are detected by many sort of like antiviral responses that are called innate immune responses in humans. In order to get siRNAs to work, it was necessary to make sure that they don't trigger that. Otherwise, they wouldn't be safe, and they would certainly not be effective. So a lot of work went into that. Fortunately, there were solutions which were using chemistry. It took a lot of chemistry to make siRNAs work so that they don't induce immune response in the organism. So they're very safe that way. They do cause some injection site reactions, a little bit of redness, but they're so safe that they are being developed for a condition of pregnancy. Imagine that. They are really, really safe. There have been thousands and thousands of doses given, and no really serious adverse effects, except for in the initial trials where they used nanoparticles to deliver, and those were quite immunogenic, and it wasn't clear at all if they were safe. Those were terminated. So yes, it took a lot of chemistry to make the molecules deliver and also deliver safely, but they're working now. And on the
stability point, very briefly.
The stability was also chemistry. One of the reasons RNA is thought to have been the primordial life form is because it does much more chemistry than DNA. RNA is much more reactive, and it largely because of this 2' hydroxyl that DNA does not have. And as a consequence, it can self-cleave, it can interact with other molecules and enzymes. To make siRNAs that were drug-like, it required removing all of those 2' hydroxyls and modifying it with things like fluorine and methyl groups and so on. So yeah, the siRNAs that are now working as drugs are really stable, which it explains why there's such a long duration of effect from a single dose.
Thank you, but I also wanted to know how feasible are these drugs, because they have to be administered every now and then, yes? I mean, I'm sorry if I'm...
How feasible? I mean, they're already approved and they're working, so they're clearly feasible, and they work really well. So I think the answer, I'm trying not to understand your question.
But they have to. You were pointing out in previous sessions that the administration actually can be quite spaced, that they have very long-lasting effect. So.
Right. So part of the, really I'd say a surprise is the durability of the knockdown, and what we think is happening is what we know is happening when you give a dose of siRNA, like into the brain, for example. It's infused into the cerebral spinal fluid through a lumbar puncture, and when you look inside the brain, let's say of a mouse, and you look at where the RNA molecules are located, what you see is that every single cell, every neuron and astrocyte and glia in the whole brain is packed full of siRNA, and it's inside these endocytic vesicles where it's probably sequestered away from the argonauts. It can't function right away, but it gradually is leaking into the cytoplasm and loading argonaut continuously over periods of months. So you get these really durable, but not an overdose. We're really lucky that they work so well. They really do work very well. And it's in humans. It's been very safe. It's going to continue to make, I think, really impactful medicine.
Thank you.
I had one last question. Why do you think siRNA is more stable than miRNA-based drugs? I mean, miRNA is more stable than siRNA, yeah?
No, microRNAs are a natural version of they load the same argonaut, so they're not more. I mean, you can make an artificial microRNA drug if you want, and you can also make a drug that blocks the activity of a microRNA. Those types of drugs are in development, and they also are chemically modified because they have to enter the body from the outside. The chemistry is required to enable delivery from the outside in. Naturally, the RNAs that are made by the cell don't need to be modified, or probably are modified, but by cellular mechanisms. So, yeah, good questions.
Thank you. Please. Thank you very much. Maybe the microphone on the stand.
Hello?
Yeah.
Thank you very much. I would like to jump to the beginning of your talk, in fact, and jump off from the small RNA and interference RNA and all this molecular stuff, and a little bit return to the pictures of the endosymbiotic theory. You know, I'm photosynthesis guy working quite a lot with this, lecturing about the endosymbiotic theory, but recently I was puzzled by the pictures which are shown in academic books and so on, because this is prekaryotic bacteria, yes, or cyanobacteria, yes, get in the endosymbiotic events to the eukaryotic cell. So my question is, do you have any thoughts about this? How the eukaryotic cell with a developed nucleus could function without good metabolism or respiring or photosynthesis, not having mitochondria or not having chloroplast before? Do you have any comments or thoughts about this?
I don't think anybody really knows how exactly the nuclear organization of a eukaryotic cell formed. There are these megaviruses that make nucleoid bodies, which are really cool, and some people believe that the origin of the eukaryotic cell was actually a megavirus infecting a sort of archaeal, bacterial cell and sequestering the genetic information inside of this nuclear envelope-like structure. But I imagine that the ancestor of the eukaryotic cell experimented with multiple different potential symbionts over millions and millions of years, and that at some point in the history, a mitochondrial symbiont sort of combo cell emerged that was so good at doing the stuff eukaryotic cells do that it sort of occupied this successful niche evolutionarily that gave rise to all eukaryotes. And that later on, eukaryotes domesticated cyanobacteria in order to bring photosynthesis. You know, photosynthesis arose on the planet 2.5 billion years ago, at least. I mean, there are algal mats that go back to like 3-point-something billion years, but we know that oxidative phosphorylation, producing oxygen, occurred beginning around 2.5 billion years ago because it caused the entire planet's climate to change dramatically. The Earth plunged into an ice age called the, what did they call it? Some jargony, nice word. But basically, the Earth was in 100 million years, it was covered with ice.
Is that snowball Earth?
It was a snowball Earth. It was the event in the history of the Earth is also when the rocks of the Earth turned red. So sedimentary rocks below that time, which can be found on the planet, were not rusted. But after that time, they were oxidized because the atmosphere prior to that had been mainly methane, and oxygen destroyed the methane. I often joke, can you imagine an organism, in this case cyanobacteria, messing with the atmosphere of the planet and practically destroying all life on the planet? I wonder how often that happens. You know, something evolves and then it wipes itself out because the Earth and the biosystems, you know, they have this really incredible dynamic.
As you say, life hasn't progressed so far, perhaps, yes. Noticing all the young people in the audience, I wondered if you had some general advice about how to, I mean, just some quick thoughts about how to approach research, how to approach a life in science.
Well, I always tell people what my mom told me: follow your passion. You know, I mean, if you position yourself because in a research area because you think you can make more money that way, that's not being true to yourself. You know, if you should do what you love and you'll be good at it. That's what my parents always said, and I really believe that. You know, and unfortunately, not all of us have that opportunity, right? I mean, there's always pragmatism. You always have to worry, well, what if I can't get a job? You know, what if I can only get a job if I work on something related to human health? You know, maybe you really would love to study ciliates and how they do their genomic remodeling every generation, which I think is a totally worthy area to study, but it's really hard to get funded for that, you know, unfortunately. And so maybe you're going to be pragmatic and say, well, I'll work on pharmaceutical development, or I'll work on HIV or something that I can get funded for. And that's a trade-off that we sometimes have to make, unfortunately. But I think if you can, you should still steal some part of your time to pursue those passions, those questions that you're curious about. On the upside, I think almost no matter what you're working on, and a lot of us, especially as trainees, as graduate students and postdocs, we don't always have complete freedom. You know, we have to do a project that's related in some way to what our advisor cares about, but you can still try to bend it in an interesting direction. You can still think about ways to make it more fun for yourself. And one of the things I was going to say earlier is the reason I get so excited about the science is because there's this whole unexplored territory that's out there, right? It's the unknown. And I think there's something that's good for the human spirit to be exploring. I think it's part of our nature. I think it's also the source of creativity. But to be encountering the unknown, a unexplored territory. It's like, I think it's just exciting, to be on the frontier. And so I really do, as hard as it is to find a path to being a scientist, maybe one where you get paid to do it, you can still be a curious person no matter what, and you can pursue that frontier of the unknown, maybe not through your own experiments, but through interacting and reading and maybe interacting with people, just to think about these kinds of questions. So anyone want to ask a question? I love it. So please, anything you want.
You've been saying about danger of information, right? And I was thinking that about the simplified model, right? It wasn't exactly showing how the thing works, and people could get wrong idea about understanding because they saw like a program, for example, and the simplified model, they didn't know how it works. Well, it was completely off. And I'm thinking, how should we teach information? How should we show the information to not give people a wrong idea? Because I see that maybe not the information is dangerous, but wrong information is dangerous, and arrogance of people who think they know how things work. And especially in current political climate, for example. And I've been thinking, you're of course authority in your discipline, right? And in like scientific field, but what could be done about teaching wrong things?
Really great comments, from such a young person too, which gives me hope, I have to say. Yeah, information, you can never really trust the data, right? The information should all be questioned. That's really a big part of the challenge, I think I naively thought information would be incredibly good, that we can have all this information at our fingertips, but it's not working out that way, right? Information is also dangerous, and it's mishandled, and we never really teach people how to handle it, and yet we're swimming in it all the time, right? I mean, a lot of you young people are on Instagram and Facebook, and I don't even know what you call the things you're on now, right? It's probably some other app. But we're inundated with information all the time, and that's why I think part of what's so beautiful about science is it values the questions, not the answers, right? It tries to disprove rather than prove, and
yes,
there's power there. But
I also feel like, for example, with NCBI, we have full access to information about genes, about how they work. We can access all this information, and like people with like degree or experience of studying a subject for several years, and a course how to use those sites, for example, and those sources, can distinguish right information from wrong more often than not, and can like get valuable core of the information. But the problem is people who are less educated and don't know how to even use those sources, not to mention understanding them and what they contain. And that's the problem, right? That like the access to information is good. The problem is that many people who even can like put in the link in the search bar, can find this source, don't understand it, and people who think they know can give them wrong idea. And I don't think that information is dangerous, but as I said, arrogance, right?
Yeah, no, I understand where you're coming from. I don't have a great answer for arrogance, unfortunately. I wish I did.
But you have been hinting again over the last few days that, exactly as you were saying, that delivering a dumbed-down message in the end doesn't serve your purpose, because people begin to expect that it's all going to be simple. And in some ways, it's about trying to spread the idea that there is complexity to these ideas, so the answer isn't simple.
Well, look at how so persistent the belief that organisms are created rather than evolved. People are, and I often encounter this in the United States, I imagine also here, because you have a very religious background in this country. These are things that, again, there's really no conflict. I mean, if you happen to believe that everything was created by God to look exactly the way it does, then you should be even more interested in how things actually work, because God created them that way. That would be so cool if God actually made everything work so perfectly. Wouldn't you want to study how God did it? I would. If I believed that that was how the living complexity arose by a creation, I would think it's even more fascinating. It'd be like a sci-fi novel to discover that it couldn't have evolved, it had to be created. I think that would be so cool to live in a universe where I know I was created by something rather than just evolved from inorganic matter into become a sentient being. I mean, that's super cool too, but I would love it if that was true. You know what I mean? So I don't know how I got onto that subject, but ideas that are wrong often persist. And, maybe everybody laughs now when somebody says, oh, the world is flat. We all know it's not. But it took a long time maybe for Galileo and people like him to get people to accept that. Maybe that's how evolution is going to work. Maybe that's how politics is going to work. Maybe that's how information handling and understanding how to handle it safely is going to work. One of the bright sides I can imagine is that it's going to be so easy to fake information that people will not trust any information anymore. So the people will know, conspiracy theorists will still thrive, I'm sure. But they won't be showing you this fake data that they like to show you, now. Anyway, we should take another question. But great ideas. I appreciate it.
Thank you very much. Please, we must stop quite soon, but
it's a great talk. Thank you very much. It's my pleasure. I have a question about the approach, because majority of this sort of novel medicines, they arose from fundamental basic biology research. And if you think about RNAi, if you think about CRISPR-Cas or the most recent novel by Carrico, where she worked for it for years to develop the mRNAs. And we know it. I mean, we are scientists, and you're scientists, and we know we have to study basic biology, fundamental biology, in order to understand anything, so that we have a potential to apply it. But how do we speak to, policymakers, politicians, biopharma, and how do we get this message across that really we need to fund the basic fundamental biology, like your very beautiful work on RNAi, and not just sort of force the translational science and essentially hope that by forcing translational science we'll suddenly figure out how to cure Alzheimer's, Parkinson's, cancers, and everything. And I see there is a huge gap and expectations.
Yeah, it's really an interesting question. How do we get society, which we all as scientists really depend on, to support our enterprise? When the returns on the investment that the taxpayer makes on somebody like me, it takes 25, 30 years before there's some tangible benefit. But and maybe it's really hard to convince them. I do think it's possible. I can tell you that in my life, my experience with medicine was really reshaped when my own daughter developed type 1 diabetes at the age of 1.5. I never wanted to be a physician because I I hated the sight of a needle. You know, I never wanted to inject or be injected, but I had to learn how to inject insulin into my own infant when she was just very little. And growing up with insulin therapy, first of all, it saved her life, but second, it creates this incredible appreciation for medicine, because this is a drug that you need to take literally every day in order to live. Without it, you will die. And so I think it's an example of a therapy that is so powerful and transformative. It's disease modifying, it gives life. And there are a lot of medicines that are like that. And diseases that affect every human being on the planet, no matter what political stripe or whatever nationality you are. I think that the hope I have is that we collectively, as a community of science and people who are supportive of science, we will continue to make good drugs and find important new discoveries that help people. And by doing that, we will win them over, because we will make medicines that save people's lives, like my daughter. And boy, does that ever bring home that appreciation for the kinds of things that AstraZeneca and others are doing to help people. I mean, if you're one of the people affected by a disease where a therapy works, you're going to be incredibly, I would imagine you would be very thankful for that. And I do think that that's the power that we have as a community. We can make the world a better place, and I think we will be valued, hopefully, in the future when we do a better job of not only doing that, but also communicating how we did it.
Thank you, Craig.
Speaking up, thank you very much.
Yeah, exactly. Communicating how the hard work that went into the kinds of discoveries that change people's lives, like insulin, which is an amazing story.
And since, we need to bring this to a close, but since you mentioned your daughter and mentioned insulin, it is perhaps worth mentioning that when you got the call from the Nobel Committee that morning in October 2006, unlike most laureates who are in the US who get that call, you weren't sleeping, or perhaps waiting by your bedside, looking at the telephone, wondering, what were you doing?
Yeah, so on that fateful evening, as I was many, many evenings, and until my daughter, who's now living on her own and taking care of her diabetes on her own and very thriving, very much so. But at that time, we were managing, my wife and I managing her care, and because I fall back to sleep easily, I had the night duty of making sure her sugar was okay. And so on that night in October, I believe it was a Sunday evening, I was up because she had a high blood sugar and I had to dose her with insulin. And so at 4:30 in the morning, I was up making sure I didn't give her too much insulin, because this is the double-edged sword of insulin therapy. Too much of it will kill you right away. Not enough of it will cause all kinds of havoc, like blindness, kidney disease, peripheral neuropathy. So you have to manage it like it's a medicine that needs intensive management, even now with pump therapy, etc. I was up checking her blood sugar, and I heard the phone ring, which is weird. I didn't expect it. I certainly wasn't expecting a call from the Nobel Committee. But when I walked back into the room, the phone rang again, and my wife said, "Don't pick it up, it's a crank call." And I said, "But, it is, the first Monday in October or whatever." And I was like, maybe they're calling about the Nobel Prize. And she got this stunned look on her face. And so I picked it up, and sure enough, it was this gentleman with a very convincing Swedish accent, Hans Jornvall, who incidentally, it turned out, I told him what I was doing, and he apologized for waking me up, and I said, no, it's okay, I was up checking my daughter's insulin. And he said, "Oh, that's interesting, because I work on insulin." And so we were talking about insulin at 4:30 a.m., and it was fun. What a wonderful surprise. And the reason we were really, really surprised is we thought microRNAs would get the prize before RNAi, and they finally gave the microRNA prize last year, which is wonderful. So now I'm really happy. And they managed to surprise everyone, including Victor and Gary, who maybe thought it would never happen.
Yeah, and they've both Victor and Gary have been saying, we saw the prize go in 2006 to Craig and Andy, and we were happy with that. We thought that's fine, that's good. And then suddenly, after all these years.
Yeah, the two pathways, as came out in the discussion, microRNA pathway and the RNA interference mechanism were related, but both independently discovered in this tiny little animal, which still has so much to teach us. Who in the room is working on C. elegans? Oh, yeah, 2. All right. Yeah, it's a great system. I'm glad that you have C. elegans researchers here. There's just so much you can do in that system. Really a great system.
I think we really have to draw it to a close, I'm afraid. It's been a really gorgeous discussion. Thank you very much, all of you, for being here, making it possible. And
Thank you.
And I don't know if you want to make any closing statement, or okay. Then, once again, it remains to thank the university very much indeed for welcoming us. And thank you, Craig, so much.
My pleasure.